dust collector and mixes it with cold water to cool it and rehydrate it. The dirt and the soil is then transported by a conveyor belt to a stacking conveyor awaiting removal. The dirt will be removed to a contamination site. A SCADA program controls the entire machine. This controls by one computer. On the screen of the computer is a diagram of each part of the machine and what it is doing, what its temperature is, how fast the drum is rotating, what belts, conveyor belts, are moving and how fast, how much fuel is being fed to the burner unit and ignited, for how long. All of these parameters are controlled by the SCADA program on the computer. Here's a picture of a backhoe loading dirt into the primary hopper. This is a small unit. You don't see the conveyor belts here of this type of desorption unit. Another method is chemical oxidation. Soils contaminated with PCE or tetrachloroethylene, which is a chlorinated solvent, can be decontaminated with chemical oxidation in a short period of time. First, the soils are analyzed for PCE levels. The contaminated soil is removed. Then clean soil is removed and stockpiled elsewhere. The contaminated dirt is then mixed with potassium permanganate, like you get for your swimming pool, which destroys the PCEs. Dirt with less than 11 milligrams per kilogram of PCEs is considered safe. Chemical oxidation is suited for small, shallow treatment areas, sandy soils, and sites accessible for an excavator. Another method, decontamination by activation of persulphate and hydrogen peroxide. This is a new experimental method that is being tested. Its control is hydrogen peroxide. The efficiency of persulphate is being analyzed. The persulphate enters the ground and is catalyzed by the natural iron found in the ground, first from the ferric iron 3 form to the ferrous iron 2 form as a catalyst, creating sulfate ions, and then in the reverse way to sulfate radicals, and then in the reverse process where the ferrous iron then catalyzes the sulfate anions to sulfate radicals by changing into ferric iron. So the iron's a catalyst. The hydrogen peroxide also reacts with the iron to form two hydroxyl radicals, which also oxidize the contaminants. The radicals do undergo other reactions that reduce the efficiency of the decontamination. The current research of this method is in trying to deduce the efficiency of the persulphate and hydrogen peroxide radicals, quantify the rate of activation in both reactions, and predict the performance during in situ or at the site chemical decontamination with benzene and decontamination reactions. The way the persulphate radical reacts with benzene in the process produces phenol. The amount of phenol is then measured. The phenol is then further reacted in the chemical process with the hydrogen peroxide, as well as the other method using the persulphate, into much less toxic chemicals, non-aromatics, organic compounds. To produce a series, okay, we went over that. This deduces the rate of reaction of the persulphate radical formation with time. The control in the experiment is the hydrogen peroxide, as I said. The efficiency of sulfate radicals yields the amount of phenol activated the efficiency of sulfate radical yield is the amount of phenol accumulated divided by the amount of persulphate decreased. The hydrogen peroxide reaction is measured and calculated in the same way. The experiment shows that although the phenol radical, the hydrogen peroxide, the hydroxyl radical activation is better, it is not more efficient. The sulfate radical is more efficient. It takes a longer period of time, however, to destroy the contaminants, but it works further down in the soil. Then biology can be used. Ex situ bioremediation. This means that the contaminated dirt is removed from the site and then treated with microorganisms and fungi which decontaminate the contaminations. This process uses fungi and or bacteria to decontaminate the dirt from hydrocarbon and volatile hydrocarbons. There are already nutrients in the dirt for the fungi and bacteria to grow. There is a few different methods, two different methods. One is biopiling. The dirt is pumped with oxygen, nutrients, and water for the fungi and bacteria to grow. Non-contaminated soil, about a foot thick, is dumped over a waterproof gravel. Aeration pipes then pump air through this dirt. Large debris is removed. The layer is then covered with a thick plastic to prevent evaporation and volatilization of harmful toxins through the process and to promote solar heating. Air handling and leaching collection systems are installed to collect and filter the effluent flows. Air is then piped into Air is then piped in to start the bioremediation process. Oxygen levels, pH, moisture, and nutrient levels are measured. Optimal levels are pH of 7, oxygen at 15%, moisture at 40 to 80% of the natural field capacity of the soil. Nutrients such as phosphorus and nitrogen may be added later if they are needed. After a few weeks to several months, this dirt is let to sit with the fungi and bacteria growing in it. The dirt is returned to its original site once laboratory analysis finds acceptable and safe limits of contaminants in the soil. The other method is composting, and there are 3 different methods of composting: closed vessels, aerobic static piles, and windrows. In vessel composting, which occurs in a closed reaction vessel, the soil is inside a metal or plastic or concrete vessel in which airflow and temperature is controlled. This is similar to a bioreactor. The air circulation is metered in by buried tubes that allow fresh air to be injected under pressure, with the exhaust being extracted through the biofilter, with temperature and moisture monitored by probes to allow optimum aerobic decomposition. Benefits of this process is that it takes up less land area and manual labor. In aerobic static piles, the dirt is placed on perforated piping, allowing for controlled aeration. This process of composting can be done outside or inside. Bacteria and or fungi decontaminate the soil after a period of time. The contaminated soil with the bacteria and fungi and mixed are mixed and plus mix in organic material like crushed up plant material, are laid out in long rows outside on the ground. And then land farming. In land farming, contaminated soil is excavated and it is placed in contaminated soil is excavated, mixed with nutrients and oxygen and moisture for the bacteria and fungi, and spread over large areas. This method is called tilling. The dirt can then be decontaminated by microorganisms and smoothed out over the land again. The plot of land is now considered decontaminated. This is a picture of an aerobic static piles facility. This is how the contaminated soil is laid out in windrows composting. And the final use of biology and decontamination, boy, it smells of gasoline down here, is in situ use of plants to reduce contaminants to safe levels. This method is less expensive than other treatments and is able to react with contamination which is far underground. According to the U.S. Environmental Protection Agency, this process consists of two different methods called bioventing and phytoremediation. In bioventing, microorganisms biodegrade organic pollutants found in the soil above the water table. The water table is the dry region above the water table is called the vadose zone. Oxygen is pumped into the dirt, increasing the aerobic ability for the microorganisms to biodegrade the contaminant.